Hello, and welcome to the lecture video for Chapter 17. I'm going to skip doing a little intro for this one and just get on to this little mini lecture. I'm hoping that this, combined with the lecture video for Chapter 18, will just kind of be two mini lectures, and then we'll be all ready to go for Exam 3. So let's talk about some of the things that influence the structure of communities. Let's bring us back to this idea of the niche. So let me remind you that the difference between a fundamental niche and a realized niche is that the fundamental niche encompasses all of the optimal environmental requirements for a species to function at its optima. Realized niche is what they're actually able to obtain in the environment. Now let's think about the idea of a niche in relation to species interaction. So in these two graphs, you have the hypothetical fundamental niche of 4 different species along an environmental gradient. And if we were to try to quantify the community at different places along this environmental gradient, you would see a shift in relative abundance at different places unless we sampled right at the environmental gradient at which the species was at its optima. And then the relative abundances at any point along that gradient are going to provide a first estimate of what the community structure might look like. Now, the actual community structure at any point along the gradient is going to be a function of the species' realized niches, which are going to be slightly different than the fundamental niche due to possible competition between species, which we're about to talk about. So the species' potential responses are modified by their interactions with the other species present. So in this fundamental niche, you're not seeing an interaction between species influencing the optimal gradient and abundance along this gradient, but you'll see a slight shift in the realized niche because of the interactions between these species, and so they're not going to perfectly match up with what you might expect in a fundamental niche. So let's talk about this null model of community idea. This assumes that the presence and abundance of the individual species found in a given community are just a result of the independent responses of each individual species. So this is very much in line with the individualistic idea of community that I talked about in the last lecture. So this is just the each species distribution is independent of other species, and it mostly has to relate to the optimal abiotic environment for that individual species and not based on species interactions. So this is the null model of a community and it's called the null because it's assuming there is no influence of community on species distributions. And like I said, interactions among species have no significant influence on community structure. So just to illustrate, this isn't really like an illustration of the null model, but it's just an illustration of looking at individual species distributions independently from their community interaction. So this is just looking at different species of birds and their relative abundance across geographic space in the U.S. So this was looking at different structures of different bird communities across the U.S. And they have, so you might imagine that these purple areas in the different parts of the map are where the species are at their optimal abiotic conditions. And you might, if you're studying each of these species individually, you might operate under this null model of community to understand their distributions. But in reality, we know that species interactions are really important. So we talked about zonation in either the last lecture or the lecture before that. Zonation typically, let's talk about how you get zonation in the first place. It generally is a result of differences in species tolerances along environmental gradients and then their interactions at those borders of the environmental gradients. So a lot of the structure that you see in zonation in communities that exhibit zonation has to do with the growth rates of the species that live there. And if the species there have higher metabolic rates or growth rates, that often restricts their ability to tolerate environmental stress. So the higher the growth rate, the more they're probably likely to be less tolerant of environmental stress, like desiccation, for instance, in these two competing species of barnacles. But this includes resource and non-resource factors. So let's look at this really classic example that I think actually might be even in your Gen Bio textbook. These are two competing species of barnacles that you can find in intertidal regions, and they show this zonation pattern where you see thalamus, I think that's how you pronounce this, thalamus at in higher tide zones, and then that would only be covered during high tide, and then you see balanus predominating closer to the ocean in areas that would be more covered by water. So without the interaction of the balanus there, though, the thalamus would be able to theoretically colonize this entire environment, but the balanus at areas that are more covered by water are able to outcompete the thalamus, so they have the fundamental niche of the thalamus or the realized niche of the thalamus has been restricted to here. And then, at least here, it seems like the realized niche and the fundamental niche of the balanus is pretty much matches up because the balanus are not tolerant of desiccation. At these higher areas, they would have more desiccation, and so that's where you see thalamus, which is a lesser competitor, be able to predominate because of environmental conditions. Okay. Here's just an example of zonation based on a soil moisture gradient and looking at different plant adaptations, so like from dry adapted to very wet adapted. So in dry adapted species, you see a lot of high allocation to roots, low allocation to leaves and stems because they're trying to absorb as much water from the soil as possible. They have lower growth rates, higher tolerance to water stress. And then the other end, at wet adapted, you see a lot of allocation into leaves and stems because there isn't limitation due to water availability. You see low tolerance to water stress. And then with this environmental gradient, you then see differences in species tolerance that create zonation patterns, and then you also see differences just based on the environmental constraints of the species that can grow in these different gradients. Here's another example from an intertidal zone, but this is looking at plants. Spartina is a genus of, I think they're called cord grasses that grow in salt marshes. They have specialized systems for ridding saline out of their system, and so they grow in like tidal marshy areas. And so you see these two Spartina zones where they outcompete these other marsh plant species. So you see Spartina alterniflora closer to the ocean. You see Spartina patens at like these medium distances from the ocean, and then like these areas that never really get covered with water. You see Juncus gerardi, and then other plant species up here. So at these upper borders, you've got competition by species that don't have to deal with salt, that are out competing Spartina in these upper regions. But then you start to see environmental constraints as you go in here. So Spartina, these two Spartina species are not great competitors when there isn't a lot of salt in the environment, but as you start to increase waterlogging and decrease the oxygen available in the soil, then they're able to outcompete other species, and that's why they dominate here. These were some experiments where they took soil from these like border zones in zonation and then grew the plant species in them with just that soil, and then they added fertilizer to it to look at the interaction. So if there's no fertilizer added and it's still got pretty low nutrition, the S. patens is a more successful competitor than alterniflora. But if you add fertilizer and nutrients to that, they're pretty much equal competitors. And then the same here in just this normal condition. The Juncus gerardi is able to outcompete the S. patens in this soil environment. But then if you fertilize it, the S. patens is a very clear competitor. So here, it's mostly competition that allows the Juncus gerardi to outcompete the S. patens. But when we are thinking about communities, it's important to think not only just about these one-to-one interactions like you see here. So like this is what the distribution of a species might be with competition. But then if you remove that competitor, then they might expand out their distribution along an environmental gradient because they no longer have that presence of competition. But in reality, species interactions are much more diffuse. So diffuse competition is the total competitive effects of a number of interspecific competitors. So diffuse competition is the total competitive effects of a number of interspecific competitors. So in this scenario, this would be the distribution of a species along an environmental gradient with no competitors. But if you just take out one competitor, so you take out the one red one, in like a more natural scenario, you're not going to see much of a shift because you have all these other competitors that a species is also competing with. You have to eliminate all significant competitors to see what you see here. So this is a more realistic scenario than this one-to-one species interaction, is that most species have more than one competitor that they're competing with. Now let's go back to food webs. Food webs are really great for studying indirect interactions, which if you're in the Monday group, we talked about this in class today. And then I have some practice questions about this that I uploaded to Schoology. Indirect interactions are interactions between species where they're not coming into direct contact with each other, as the name suggests. So an indirect interaction would be an effect of interactions between like soapberry and lynx populations, or any interaction between like these predators and these plant populations. So when you look at this graph this is with snowshoe hare being a keystone species in this food web. These are all the indirect interactions would be like the plants with, so like maybe aspen with passerine birds, white spruce with moose, and those kinds of things, because there's an intermediate species in there. So if we think about a scenario where this indirect interaction might affect the other species, now let's think about this. So if there was a decline in the white spruce population, you might see a decline in the spruce grouse population, which then would result in a decline in the moose population because the white spruce is affecting the spruce grouse. Or let's say you have, now snowshoe hare feed on a lot of different types of plants, but let's say that, here, let's pick another one. that feeds only on a few plants. So like, oh, here we go. So passerine birds feed mostly on insects and white spruce, but if they don't have their herbivorous source, so if you see a decline in white spruce, you might see a subsequent decline in passerine birds. That decline would then maybe also cause a decline in the harrier. So that would be a look at the population dynamics that might be involved in indirect interactions. Let's go back to the concept of keystone predation too. So, or keystone species. So keystone predation is a variation on this idea of a keystone species, and this is when a predator enhances one or more less competitive species by reducing the abundance of more competitive species. So a predator comes in, it eats more competitive species, and then other species that compete with that species take over. So this would be another example of an indirect interaction. So in this example here, this starfish feeds on lots of different things, and when you remove the starfish from the community, then you have reduced the diversity of the prey as a result of increased competition among the prey items. So the starfish actually increases the diversity by keeping the populations of its prey items low enough that they don't outcompete each other. So you remove the starfish, then you maybe get like a boost in gooseneck barnacles species, and then they outcompete whelks and limpets, and so you see a decline in these species. So that's how keystone predation works. You can also have apparent competition. This is when two species that do not compete with each other for limited resources affect each other indirectly by being prey for the same predator. So in this aphid example, grass and nettle aphid use different host plants. So you have grass aphids eat on grass, nettle aphids probably eat stinging nettle. But both of them are preyed upon by ladybugs. So if you increase grass productivity through fertilization, you get an increase in the grass aphid populations, you get an increase in the ladybugs, and then you see a decrease in the nettle aphid populations. But with the opposite, if you have, you're not fertilizing the grass, so in this case the nettle is more reproductive, then you'd see a boom in the nettle aphids. Oh, actually, no, you don't see that at all. You really only see it here because, sorry, you see 2 ladybugs, not 4 ladybugs. So only here. So this is apparent competition between the grass and nettle aphid. So I'll explain that again. So in this plot where the grass is fertilized, you see an increase in the grass aphids. Ladybugs are eating those grass aphids, so you see an increase in the ladybug population. That increase in the ladybug population then causes a decrease in the nettle aphid. So that's apparent competition because even though they don't feed on the same host plants, an increase in their population causes an increase in the predator, which then suppresses the other population. Now let's talk about indirect commensalism and indirect mutualism. So indirect commensalism is just basically any indirect interaction between two species that can qualify as commensalism. So one benefits, the other is not affected at all. And then indirect mutualism is when one species benefits the other species but doesn't actually directly interact with it. So in this example that we have here, which I have a practice question that's kind of similar to this in the thing that I uploaded for exam review. A positive means there's a positive impact. Negative means they're negative. So Daphnia pulex positively impact larval salamander populations because the larvae eat the Daphnia. The salamanders feed on the Daphnia, so they negatively impact it. The Daphnia pulex and Daphnia rosea are competing species, so they negatively impact each other. Midge larvae feed on Daphnia rosea, so Daphnia increase midge larvae midge larvae decrease Daphnia rosa. And then what you see by these dotted lines, these are indirect interactions between the salamander, Daphnia rosa, and the salamander in the midge larva. Now what's going on here is that because of this competition between Daphnia pulex and Daphnia rosea, when larval salamanders are present, they feed the Daphnia pulex. The Daphnia rosea have a reduction in their competitors, so their populations increase. And then that increase in the Daphnia rosea population then causes an increase in the midge larva. So larval salamanders, by causing a decrease in the Daphnia pulex population, are benefiting Daphnia rosea and the midge larva. That's what's illustrated here. So if you were going to do an experiment designed to test indirect interactions, you can really only do these in controlled experiments involving the manipulations of the species. So like if you wanted to figure out what these indirect interactions were of the larval salamander on Daphnia rosea and the midge larva, you would grow these 4 species in an environment together, and then you would eliminate the larval salamander from the equation and see what happens to population sizes of the midge larva and the Daphnia rosea. Likely what would happen is that if you remove the salamander from the experiment, your hypothesis would be that Daphnia pulex populations would increase. They would outcompete Daphnia rosea, so you'd see a decrease in this population, which would then cause a subsequent decrease in the midge larva. So basically, if you want to test indirect interactions between species, you have a control with all of them, and then you have experimental manipulations where you either eliminate one of the species from the interactions or you lower its numbers in comparison to the control. So be prepared to be able to design an experiment when presented with an example on the exam, and then hypothesize what the interactions might look like based on the information you're given. In food webs, when we look at food webs, this is that snowshoe hare example again. These can tell us a little bit about what's controlling population dynamics and community structure in these environments. So bottom-up control is when the productivity and abundance of populations at any given trophic level, so predator, intermediate, basal, are controlled or limited by the productivity and abundance of populations in the trophic level below them. So if this were a bottom-up system, this would be this level being controlled by this level being controlled by this level. Now, on the other hand, you can have top-down control, and this is when predator populations control the abundance of prey species. So this would be the lynx controlling the populations of the snowshoe hare and the ground squirrel. So top down, bottom up. So top down, bottom up. And then you can also have a trophic cascade, which is when a predator in a food web suppresses the abundance of their prey, such that it increases the abundance of the next trophic level. So this would be like lynx eating so many snowshoe hares to a point that it increases the diversity of the plants that are in that environment. And I've uploaded a video onto Schoology I'd like you to watch about trophic cascade involving coyotes in Yellowstone. It's a really, really cool experiment where coyotes were hunted to near extinction in the park. They were reintroduced, and you saw enormous changes in the entire plant structure of Yosemite because of the reintroduction of these coyotes, which then caused a trophic cascade. Now, environmental heterogeneity can also influence community diversity. So this was, you should recognize this from exam 1. This was that grassland study looking at the diversity of nest building and cuckoo bees. And when you have more heterogeneous landscapes of vegetation, so more diversity in the landscape and the types of vegetation, That's also going to influence the diversity of the animal species that the vegetation depend on. So the more vegetation you have, the more you add to the community species interactions of animals that depend on those plants. And then if you increase the diversity of food resources in a community, you can also likewise potentially increase the diversity of the animals that consume them. So a general rule for like bee conservation is that if you plant it, they will come. I think that's Rich Hatfield from the Xerces Society's like motto is that if you plant a really diverse native flower source, then diverse communities of bees will likely follow with the plants that you're introducing. So likely in this study, looking at bee species richness, as habitat area increased, so did the diversity of plants in that area, and then so too did the diversity of bee species that were feeding on those plants. And that's it. This is a little shorty. So stay tuned for another little shorty for chapter 18, and then we'll be done for exam 3. Okay, bye-bye.